Good morning to all my dear students. Welcome to our zoology online class. Okay, my dear students, in the last class we started the 7th lesson. Okay, human health and disease. And in the last class we studied about two important 5-mark questions. That means 8 bacterial diseases and 8 viral diseases. Already I told you this is very important 5-mark question. Okay, my dear students. So in the last class we studied about bacterial disease and viral diseases. So today we are going to study about some protozoan disease. Some protozoan disease. So listen very carefully, dear friends. Okay. Today our first disease. Today we are going to study about some protozoan disease only. So the first disease is African sleeping sickness. African sleeping sickness. This African sleeping sickness is caused by a protozoan species. Name of the protozoan species is called Trypanosoma species. Trypanosoma species. So these species are only responsible for some different types of African sleeping sickness disease. Understand? Then this disease is carried or transmitted by a vector. Okay, one flies act as a vector. Name of that vector is called tsetse flies. Okay, so these flies only carrying this Plasmodium species and spreading this protozoan disease, African sleeping sickness. Understand a few? So now we complete this. Now we have studied the causative organism and vector name of this African sleeping sickness. Understood, my dear students? Okay. Today now we are going to study about 3 types of African sleeping sickness. See, the first one is Central African sleeping sickness. Second one is East African sleeping sickness, and the third one is American trypanosomasis. So these are the some types of African sleeping sickness diseases. Okay, the first one is Central African sickness. This disorder is, this disease is caused by a Trypanosoma species. Name of that species is called Trypanosoma gambensis. Okay, so this is the causative organism of this Central African sickness. Then, so this disease is transmitted by a flies. Name of that fly is called Glossinia palpalis. So this is act as a vector. So this insect, this flies only spreading this Central African sickness. Then second one is East African sickness. Okay, so this East African sleeping sickness is caused by a protozoan species. Name of that protozoan species is called Trypanosoma rhodesiensis. Okay, so this is a causative organism of this East African sleeping sickness. Then the vector Glossinia morstans. Okay, so this vector only spreading this East African sleeping sickness. Understand, my dear students? Then last one is American trypanosomasis. Okay, American trypanosomasis. So this is also one of the example for what this is African sleeping sickness. Understand a few? So now we completed 3 types of African sleeping sickness. First one is Central African sickness, second one is East African sickness, and the third one is American trypanosomasis. Then the second disease, second example for the protozoan disease is Kala Asar. This Kala Asar is caused by one protozoan species. Name of that protozoan species is called Leishmania donovani. Okay, Leishmania donovani, and this disease is transmitted or spread by a flies. Name of that fly is called Ferifortums. Okay, Ferifortums is the vector of this Kala Asar. And maximum this Kala Asar disease affecting our internal organs like the bone marrow, blood vessels, lymph node, spleen, like these organs are maximum affected because of this Kala Asar disease. Understand? And if anyone is affected with this Kala Asar disease, we can see some symptoms, particularly weight loss. So weight loss, anemia meaning is a disease related to blood. Okay, the disease affecting the blood. Then fever, enlargement of spleen and liver. These are the symptoms of this Kala Asar disease. Understand a few? So now we have completed 2 important diseases. The first protozoan disease is African sleeping sickness, and the second disease is this Kala Asar. Okay, then. Then the third disease is, amoebiasis. Okay, the third one is amoebiasis. This amoebiasis is also known as amoebic dysentery. Amoebiasis disease is otherwise called amoebic dysentery. And this amoebiasis disease is caused by one protozoan species. Name of that protozoan species is called Entamoeba crystallitica. So Entamoeba crystallitica is the causative organism of this amoebic dysentery or amoebiasis disease. Understand a few? Then maximum this microorganism, this protozoan species living inside the large intestine of our body, living inside the large intestine of the body. Maximum they are eating the food particles present in our large intestine and the bacteria present inside our large intestine. So the food particles present inside the large intestine and the bacteria present in the large intestines are eaten by this Entamoeba crystallitica parasites. Understand a few? Then finally they are rupturing the intestine wall, large intestine wall. Okay. Then the infective stage of this Entamoeba crystallitica is called trophozoite. So trophozoite is this stage Entamoeba crystallitica only causing this amoebic dysentery to human body. Understand? Then this Entamoeba crystallica reaching this trophozoite stage, they release a enzyme. Name of that enzyme is called histolytic enzyme. Name of the enzyme is called histolytic enzyme. So maximum they are living inside of a large intestine, eating the food and bacteria present in the large intestine. Then finally they are releasing one enzyme. Name of that enzyme is called histolytic enzyme. Understand a few? Then the symptoms of this amoebic dysentery or amoebiasis disease. The first one is ulcer. Okay, so this microorganism eating the wall of the large intestine. So finally some ulcer or wound is formed in the large intestine. So ulcerization or ulcer is the first main symptom of this amoebiasis disease. Then second one is bleeding, internal bleeding. Third one is abdominal pain. Then stool with excess mucus. Here stool means motion. Okay, so human waste with more amount of mucus. Mucus means in Tamil we can say check. Understand a few? Then the cause is the vector of this disease. So this disease is transmitted by a fly, the ordinary houseflies. So the ordinary houseflies only acting as a vector or carrier of this amoebiasis disease. Understand a few measurements, friends? So now we have completed 3 diseases. First disease African sleeping sickness, second one cholera, and now we have completed amoebiasis. Understand a few? Then the fourth protozoan disease is malaria. Fourth protozoan disease is malaria. See, maximum the malarial fever is caused by different varieties of protozoan species. Okay, so the name of this protozoan species is called Plasmodium. So different types of Plasmodium species causing different types of malarial fever to human body. For example, Plasmodium vivax species. Okay, causing one malaria disease, then Plasmodium ovale, then Plasmodium malariae, then Plasmodium falciparum. So these are some of the example for Plasmodium species, and all these species causing different types of malarial fever. Then next point, this malaria also transmitted by a vector. Female Anopheles mosquito is acting as a carrier or vector of this malaria disease. So female Anopheles mosquito is acting as a vector of this malaria disease. Understand, my dear students? Okay. Now we are going to study about one important 5-mark question. Very important 5-mark question. The life cycle of Plasmodium. Okay, the life cycle of Plasmodium. Listen very carefully, my dear students, because it is very important for both science group and biology students. Okay, say the Plasmodium is a digenic parasite. Digenic parasite means living in 2 host organisms' body. Female Anopheles mosquito is the primary host of this Plasmodium species, and the human body is the secondary host of this Plasmodium species. So this Plasmodium species is a digenic parasite. Then the life cycle of this Plasmodium species has 3 important stages or 3 important phases. Okay, the first phase or the first stage is called schizogony. First stage is called schizogony. The second one is called gamoscony, and the last one is sporogony. Okay, so schizogony, gamogony, and sporogony. So these are the three phases or three stages of in the life cycle of the Plasmodium species. Understand, my dear students? Then see, actually this Plasmodium species transmitted to a healthy person by a infected female Anopheles mosquito. So when a infected female Anopheles mosquito biting a healthy person, okay, bite a healthy person, the trophozoite which are present inside the salivary gland of the female Anopheles mosquito is injected in the human body or injected in the human blood. Okay, so the infected female Anopheles mosquito, okay, infected female Anopheles mosquito injecting or releasing the sporozoite, okay, the sporozoite inside the human blood. Understand of you? Then, when it reaches the human blood, this sporozoite moving to the RBC or entering inside the human RBC. Then from the human RBC, they penetrate or enter inside the hepatic cells of the liver. They penetrate or enter inside the hepatic cells of the liver. So, first the sporozoite is injected in the human blood from the salivary gland of a infected female Anopheles mosquito. Then this sporozoite is entering inside the RBC of the human blood. Then from the RBC it is entering inside the hepatic cells of the liver. So hope you all understood all this point. Then the next point is inside the liver. This Plasmodium species, this sporozoite, okay, this sporozoite stage involving a asexual reproduction, involving in a asexual fission reproduction, and produce the next stage. It is called merozoite. Understand of you? So when it is entered inside the liver, immediately it is involved, this sporozoite involved in the asexual fission reproduction. Then at the end of this fission reproduction, they produce the next stage. It is called merozoite. Then this merozoite is penetrate or enter inside the RBC. Enter inside the RBC. Understand of you? Then inside the RBC, this merozoite is growing in its size. And this merozoite stage getting a large central vacuole and pushing them in one side of the cytoplasm. Listen very carefully. So the merozoite is penetrating inside the RBC. After reaching the RBC, they increase in their size. They are growing in their size, and finally they are getting a large vacuole. Understand of you? Then after getting this large vacuole, now this stage is called signet ring stage. So I hope you all understood this point. After reaching the RBC, this merozoite enlarging their size and getting a large vacuole, and now it is converted into next stage. This stage is called signet ring stage. Now this merozoite, the nucleus present in this merozoite is divide and produce the next stage. It is called schizone stage. So the nucleus present inside the trophozoite is dividing in their number and producing schizone. Now the schizone getting a yellow brown color granules. So large number of yellow brown color granules are formed inside the schizone stage. Okay. Now these yellow color granules are called Scoopner's granules. So listen very carefully. So the nucleus of the trophozoite divide and producing schizone. This schizone consists of large amount of yellow brown color granules, and these granules are called snuffers backwards. Understand of you? Understand? Then this schizoid, now this schizoid dividing in their number and produce large number of merozoites large number of merozoites. Understand of you? So now all these processes takes place inside the RBC. All these processes takes place inside the RBC. So after producing this merozoite, the RBC is lysed. RBC is breakdown. Understand of you? Immediately from the RBC, more amount of merozoite and a toxin is released in the blood. Okay. So name of the toxin is called hemocyte. Understand of you? So after this erythrocytic cycle is over, the erythrocyte or the RBC is broken or lysis. Then from the RBC, more amount of merozoite and one toxin is released. Here the name of the toxin is called hemocyan. It's called hemocyan. Then this up to this stage it is called erythrocytic cycle. So this erythrocytic cycle takes minimum 48 to maximum 72 hours. Okay, so one erythrocytic cycle takes 48 to 72 hours. Understand of you? Then some malarial species taking more time. Okay, so based on the species of Plasmodium, this erythrocytic cycle's duration is increasing. So minimum 48 hours. Minimum 48 hours. You understood, my dear students? Then the next point is, see, now merozoites and chemocyan toxins are released inside the blood. Isn't it? Now this merozoite is differentiated into two gametocytes. It's differentiated into macrogametocyte and microgametocytes. So the merozoite and the chemocyan toxin both are present in the blood of the human beings. Okay, so after reaching the blood, this merozoite is differentiated into macrogametocyte and microgametocyte. Understand of you? When a female Anopheles mosquito sucks these micro and macrogametocyte, automatically it is entering inside the body of the female Anopheles mosquito. Then after reaching the female Anopheles mosquito's body, this macrogametocyte is changed into male gamete and microgametocyte is changed into female gamete. So this is happening inside the body of the female Anopheles mosquito. Inside the body of the female Anopheles mosquito. Understand of you? So after reaching this female Anopheles mosquito body, this macrogametocyte converted into male gamete and the microgametocyte converted into female gamete. Now both gamete are fusing or joining to form a diploid zygote. Okay, name of this zygote is called ookinete. It's called a ookinete. So I hope you understood. So after reaching the mosquito's body, both gametocytes, male macrogametocyte and female microgametocyte, both are joining or fusing together and forming a diploid zygote. This diploid zygote name is called ookinete. Now this ookinete migrate or moving to the wall. Okay, to the wall of the large intestine. Sorry, it is moving inside the wall. Understand of you? So after reaching the wall of the gut, so this ookinete is converted into oocytes. Ookinete is converted into or changed into oocytes. Listen very carefully, my dear students. So the both microgametocyte and the macrogametocyte both are fused together and forming a ookinete. Then this ookinete is moving to the wall of the gut or wall of the intestine. After reaching the wall of the intestine, it is changed into oocytes. It is changed into oocytes. Understand of you? Now this oocyte develop into sporozoite. So inside the gut wall, this oocyte is changed into or developed into sporozoite. Understand of you? This sporozoite is again moving to the salivary gland of the female Anopheles mosquito. Is moving to the salivary gland of the female Anopheles mosquito. Understand? When this female Anopheles mosquito bite a healthy person, automatically this sporozoite is released from the salivary gland of the mosquito to human body. Okay, now one cycle is completed and next cycle is started. Understand of you? So this is the life cycle of this plasmodia. Understand, my dear friends? So it's very important question and very important 5 mark questions. Understand? Then maximum this malarial species, okay, the plasmodium species affecting the RBC, spleen and some visceral organs. Affecting the RBC, spleen and some visceral organs. Understand a few? Then maximum the incubation days or the incubation day duration of this malaria is 12 days. The incubation period of the malarial fever is 12 days. Understand a few? Then see, after transferring or after spreading these plasmodium species inside the body of a healthy person, they will feel some symptoms. Maximum the symptoms are headache, okay, headache, high fever and muscle pain. So these are the common symptoms after getting these plasmodium species inside the body of the human body. Understand a few? Understand a few? Then this hemocyan toxin, already I told you. Okay, the hemocyan toxin is released inside the blood, and the RBC based are released inside the blood. Immediately the patient feels some severe symptoms like shivering, chill, high fever, high sweating. Okay, so these are the symptoms when the patient feel when this hemocyan toxin and the erythrocyte debris are mixed, erythrocyte based are mixed inside the blood. Understand of you? Then when this toxin, hemocyan toxin is released inside the blood, immediately they release 2 more important factors. One factor name is called the tumor necrosis factor and the second factor is called interleukin. Okay, so after this hemocyan toxin is released inside the blood, these 2 factors are also released. What are the factors I said? Tumor necrosis factor and interleukin. Understand, my dear students? Then prevention. See, the one and only way to prevent this disease is killing the vector. Here vector, the female anaphylaxis mosquito. So we have to kill the mosquito species. So this is the major prevention methods. Understand? And maximum this mosquito species laying their egg inside the water bodies. Okay, inside the stagnant water bodies. So to eradicate the mosquito species, oil is sprayed over these water bodies. Oil is applied over these water bodies. Okay. So when we apply the oil on the outer surface of the water bodies, the egg which are laid inside the water bodies is cast into larva. This larva coming outside for breathing purpose to get the atmospheric oxygen. Understand? So for breathing purpose, the larva of this mosquito is moving from the water body to outer surface of that, means to the atmosphere, to reach the atmosphere to receive the atmospheric air. So when we apply oil on these water bodies, they cannot penetrate the oil, they cannot get the atmospheric oxygen. So automatically the larva of the mosquito species is destroyed. Okay, so this is one of the way. Then second one is culturing some fishes inside the water bodies, Gambusia fishes. So these fishes can eat the larva of the mosquito. So we have to culture these fishes in the water body. So this is the second prevention method. Understand of you? The third one method is spraying one useful bacteria, Bacillus thuringiensis. Okay, so when we apply this bacterial species in the water bodies, when we spray this bacterial species in the water bodies, automatically they will destroy the larva of the mosquito. They will destroy the larva of the mosquito. So these bacterial species will not cause any danger to any other living organisms of the water body. Understand a few? Then next prevention method is using mosquito net. Okay, we have to avoid biting the mosquitoes. Okay. Understand? So our house, windows, doors, everywhere this whatever. So we have to fix a mosquito net to avoid the mosquito. Understand of you? So these are the prevention methods. Understand, my dear students? Then see the last point about this malaria. This malaria disease. For World Health Organization introduced one program, already introduced one program. Name of that program is called Mosquito Eradication Program. Mosquito Eradication Program. But this program was not successful. Because killing of this mosquito is one of the very difficult tasks. Okay, so we cannot kill the mosquito species easily. We cannot eradicate all the mosquito species easily. Understand? So this program was failed or not successful. Then, moreover, this Plasmodium species getting higher resistant power against an insecticide and pesticide. Example, DDT. Okay, so the Plasmodium species of this malaria species can resist this, resist against this DDT like this pesticide and insect. So killing these Plasmodium is little difficult. Understand, my students? So dear students, today we have completed some protozoan diseases. The first disease, African sleeping sickness. Second one, Kalasa. Third one, amoebiasis. Now we have completed the malarial disease also. Understand? And here we have studied one important 5-mark question, the life cycle of Plasmodium. Understand up to? So all study well and write this answer. After studying, you write the answer and send to me through my WhatsApp number. I will correct it. Okay? Thank you. We will meet in the next class.